Okay, hi everybody. Welcome to the February, almost Mardi Gras edition of Micro Seminar. We have reproductive destruction, or at least moderation, in Wolbachia, coming to you from Irene Newton. She's a graduate of Swarthmore, so our second Swarthmore alum in a row. And then she did her postdoc at Harvard with Colleen Kavanagh.
Oh, that was my PhD.
Sorry, PhD. I'm skipping down the line here. And then her postdoc with the father of pathogenesis, which I don't even know what that means, but his name is Ralph Isberg at Tufts. So without further ado, actually, one thing. Irene is welcome to take questions, welcoming questions all throughout the micro seminar today. So if you make sure you tweet any questions to the hashtag #USeminar. And I'll catch them, and then if they're real and pertinent or important, Irene has asked that I interrupt her so that she can take them on the fly. So go ahead and fire your questions all throughout, and we'll take questions at the end too. But without further ado, Irene, take it away.
All right, so I'm going to attempt to start this screen share process. See if I can get this going. And hopefully you'll let me know if you can see this.
I can see it. It's just get it all the way up to full screen.
I did.
So right now we see it sort of in the presenter version.
Oh, you see it in presenter mode. Interesting. Okay. Let me try a different way of sharing it. And try now.
Perfect.
Okay, excellent. So I'm excited to talk to you today about our work on Wolbachia, and its prime, interesting reproductive manipulations just in time for Valentine's Day. And I thought I'd start by showing you a little bit of a little portrait of Wolbachia, if you would. So what you see here on this introductory slide is Wolbachia stained with the eubacterial probe with fish. And that's in green, and then you can see the nuclei of the host in blue, and this is a reproductive tissue in Drosophila melanogaster called the germarium and the egg chambers. And what you can see from this image is hopefully a lot of green staining, so you can know that Wolbachia really densely colonizes reproductive tissues. And also you might notice, and I've kind of highlighted in the little white square here, that Wolbachia are localizing to what will be the oocyte in the next generation. So they've figured out a way to colonize the next generation even before that next generation is laid, before that egg is actually even fertilized. And this is what Wolbachia is really good at. It's a reproductive parasite, and it's maternally transmitted. And I'll be telling you about the work from our lab that's highlighted some interesting interactions between Wolbachia and the host cytoskeleton, primarily actin. Right, so Wolbachia, if you know anything about it, you've probably heard about the different reproductive manipulations that it induces in various insect hosts. So on the extreme end of things, we have these phenotypes like feminization, where male offspring are turned female, parthenogenesis induction, where females produce effectively clones of themselves, male killing, where Wolbachia actually kill male offspring, and then the most common phenotype, which is cytoplasmic incompatibility, which can be thought of as just sperm-egg incompatibility, where Wolbachia infected males cannot mate with uninfected females. And so each of these strategies, although they seem bizarre and disparate, really forward Wolbachia's goal, which is to increase its prevalence in a population and increase the number of females who can spread Wolbachia through that population. So just to highlight who Wolbachia is, obviously the reproductive parasite that manipulates these insect-sex ratios. I think of it also as the queen of the alpha protease. So it infects 40 to 60% of insects on the planet. And that's a staggeringly large number of different species that are infected, making Wolbachia likely the most prevalent intracellular infection on planet Earth. That is not counting the mitochondria. It is also the infection that keeps on giving. I showed you the picture of Wolbachia infecting the next generation even before that next generation matures and unlike these pathogenic acute infections that people are familiar with, Wolbachia is an intracellular infection that is persistent and is maternally transmitted. So it is the infection that keeps on giving every generation. And it might be one answer to the control of infectious disease vectors. So recently, maybe it's not so recent now, back in 2008, it was published that Wolbachia-infected insects actually prevent the replication of RNA viruses. So let me explain a little bit about how this would work. So let's say we have our mosquito vector, our Aedes aegypti here, and what people are doing is infecting it with Wolbachia. Then when that mosquito goes on to take a blood meal from an infected person, the dengue virus cannot replicate within the mosquito. So when the mosquito bites another person, it does not transmit the virus. So in this way, you are preventing the transmission of this RNA virus by manipulating the vector, specifically through infection with Wolbachia. And this is becoming more relevant these days as global warming is altering the range of these different disease vectors. And if you keep up on the literature or on the tweets that are out there with regards to infections seen in different regions, you might notice that chikungunya was popping up in the Caribbean and also in Florida. So it's something that will start to affect more and more Americans, and obviously is affecting large portions of the world. All right, so as part of this talk, I'm going to focus on two different projects that have come out of our lab recently. The first is identifying host genetic determinants necessary for Wolbachia to replicate and be transmitted to the next generation. And the second part of the talk will be our 4 genetics idea for Wolbachia in an intracellular symbiote. So I will present to you the data that we have and let you come up with your own conclusions as to whether or not we can actually use this to generate mutants in Wolbachia and characterize their phenotypes.
When last night?
Shut up, seriously.
Back on track.
Who knows?
Okay. So we'll start with the following premise, that manipulation of actin is a requirement for any bacterium that lives intracellularly. So Wolbachia live within host-derived vacuoles, and you can see in this electron micrograph here, Wolbachia is surrounded by these really nice membranes. So it's found predominantly in these host-derived vacuoles. And in order to enter and exit these non-phagocytes, Wolbachia must be able to transit the cell membrane and be internalized. And this is not an uncommon strategy for bacteria. So I'll show you this nice picture from Jorge Galan's group showing Salmonella with the classic membrane ruffles that are actually being promoted by Salmonella itself. So through the injection of type 3 effectors, Salmonella is promoting uptake in these epithelial cells, producing these beautiful structures and allowing the bacterium to invade the intracellular niche. So it's likely the case that Wolbachia, also an intracellular bacterium, has a similar strategy for manipulating host actin. Now, previously, lots of research had been published focusing on Wolbachia and the microtubule cytoskeleton. So what I'm showing you here is if you take a female Drosophila and you dissect the ovaries, you'll see these two kind of bunches of grapes. Is what they look like, this tissue. And if you tease apart those bunches of grapes, you will find these little beads on a string, these ovarioles or egg chambers, where you can see different stages of Drosophila oogenesis, one next to another, so these progressive stages of development, starting with the germarium, which is home to the stem cells and the stem cell niches, and going all the way down to the mature egg before it's laid by the female. And what's nice about this system for us is that it allows us to look at localization and titer of Wolbachia throughout the development of the oocyte. And what other people had shown previously was that very early on, during the process of oogenesis, Wolbachia localizes to what's going to be the oocyte. And it does this in a kind of a curious anterior localization. And you may have noticed that from our images that I showed you in that first slide. And as it turns out, microtubules and motors are required for this anterior localization. Later on, during oogenesis, Wolbachia required kinesin 1 for another kind of characteristic Wolbachia localization, which is localization to the pole in these later stages of development. So it had been known that Wolbachia used the microtubule cytoskeleton to achieve these localizations, and that these localizations were likely important for transmission, although that hadn't yet been shown. But was there any evidence that Wolbachia used actin? And indeed, there was. For example, you can take a Wolbachia preparation, so although we can't culture them outside of cells, we can culture them in cell lines, and we can actually prepare Wolbachia fractions from infected individuals. And you can take that and inject it into uninfected fly, for example, shown here. And if you inject it into the abdomen, the Wolbachia actually transit to this special tissue, the germarium, so they make it to the germline, traversing multiple tissue types, multiple cell types, and being engulfed by these non-phagocytic cells. So that suggests that Wolbachia certainly have the ability to be able to manipulate the host cytoskeleton and actin. And in a subsequent publication from a different group, it was shown that this characteristic localization in this tissue, the germarium, where the germline is derived from the female, that this localization is more congruent with the Wolbachia phylogeny than with the host phylogeny, suggesting that there's some Wolbachia-specific factor that's behind this localization. All right, so with that background in mind, we had the question, do Wolbachia require actin for infection or for persistence within the host? And the route we took to answer was using a genetic test in Drosophila melanogaster. So I should mention that Wolbachia is a natural infection of Drosophila melanogaster, and that most of the fruit flies that you'd encounter on bananas or in your house are likely infected with Wolbachia, and that I am at Indiana University in Bloomington, which is home of the Bloomington Drosophila Stock Center, all of these genetic stocks that people have created and deposited and that are maintained up on the 5th floor, and about a third of these stocks are infected with Wolbachia. And so we began this project by looking at 3 different Drosophila stocks, showing you here 2 that are mutant in profilin, which is a protein that binds to actin monomers and regulates filament formation by bringing these monomers to the nascent filament, and also by stabilizing G-actin. Then we also looked at vilin, which is an actin regulatory protein that bundles actin filaments. So you can think of both of these actin regulatory proteins as acting on the cytoskeleton, the actin cytoskeleton, and regulating the amount of G and F actin that there is available in the cell. So as a first pass, what we did was check whether or not these particular stocks were infected with Wolbachia. So what you see here is the result of a Western blot where we've probed using a Wolbachia-specific antibody, anti-WSP, and you can see our infected line here, WMEL. You see a nice, clear, and distinct band. But in our stocks, these mutant stocks, we didn't see anything. So we knew these were not infected with Wolbachia. And so we took a strategy to cross them to infected females to then introduce Wolbachia into these backgrounds. This is probably the easiest way to set up a Wolbachia infection in your fly stocks. So here are our crosses. We took Wolbachia-infected females with white eyes. We crossed them to males of this stock, and then in the first generation recovered the progeny that had the mutation in a heterozygous condition. These females were crossed back to the paternal line, producing the second generation. And so our hope was to identify whether or not there were, maybe Wolbachia localization differences or patterns that would change in this mutant background. And instead we saw a very interesting phenotype. So what I'm showing you here is the result of a PCR amplification of a Wolbachia-specific gene within our maternal wild-type stock. And here are our mutant stocks that were uninfected. So you can see the band that's characteristic of Wolbachia here, and our blanks. And in the first generation, we saw a reduction, qualitative reduction in Wolbachia, and then the inability to detect Wolbachia, at least by this amplification in some of these lines. And then Wolbachia seemed to be absent in the second generation. So again, this was very curious, because if you know anything about Wolbachia, you know that it's very good at maternal transmission. This is kind of what Wolbachia has evolved to do, is to be maternally transmitted and to infect the reproductive tract and manipulate reproduction. So this said to us that the regulation of actin might be important in this phenomena. And we confirmed this phenotype using both quantitative PCR, which I'm showing you here. So again, looking at our control flies, and then the first generation of these heterozygous mutant flies. In addition, some deficiency stocks that we added in. So these are stocks from the Bloomington Drosophila Stock Center that have deletions of chromosomal regions in the same area as these loci of interest. So again, just pointing to the fact that these loci are important for this particular phenotype in a completely different genetic background than the first set of alleles that we had looked at. Right, and then we also recapitulated this phenotype using our Western blot approach to looking at Wolbachia titer. So we have our control flies here and our first generation and second generation crosses. Again, showing this reduction in total Wolbachia load as you passage Wolbachia in these mutant backgrounds. And this is a variable phenotype, so if you look across individual F2 progeny in this particular background, you'll see significant reductions, or you'll see what look like relatively less severe reductions across the board, but certainly less than what we see in our control flies, where we always see a very large amount of Wolbachia, at least based on the anti-WSP probing on this Western blot. So that led us to the following question: what is going on in these first-generation progeny that result in the loss of Wolbachia, or this inefficient transmission to the second generation? These progeny are the first example of Wolbachia coming into contact with these alleles, these mutant alleles. So we wanted to look in that germarium, in that structure where Wolbachia colonize the earliest stages of oogenesis. So what I'm showing you here is our wild-type control flies. So first staining the actin cytoskeleton with phalloidin, and then staining Wolbachia with this anti-HSP60 antibody. So what I hope you can appreciate is that Wolbachia densely colonized this region of the germarium. This is the so-called region 2, where the somatic stem cell niche is and where Wolbachia are thought to make their home in the fly. And then I hope you also see this kind of characteristic localization of Wolbachia to what is presumed to be the early oocyte. And I'm going to show you some comparisons here with our first-generation mutant flies so you can see what the phenotype was. So in our quail mutants and in our 2 chickadee mutants, we saw this very distinct Wolbachia aggregates in this tissue, as opposed to the more diffuse labeling of Wolbachia within this region. And we also saw a relatively less staining or diminished staining later on in later stages of oogenesis. So if I should mention that these aggregates are extremely fluorescently intense. So if you were to look at these raw images, you would notice quite obviously that they are kind of aberrantly intense in the fluorescence that we're detecting here. And we can quantify the difference that we observed at these different backgrounds by measuring Wolbachia fluorescence intensity in the entire germarium in blue, in region 2 in green, and in the earliest stages of oogenesis in red. And in our control flies, this is the pattern that we observed, so a fair amount of total staining, and then lots of staining in the earliest stages of oogenesis, and some in region 2 as well. And we saw a statistically significant reduction across the board in each of our first-generation mutant flies. So less staining overall, and then also you might notice this interesting pattern switch, where instead of having lots of staining in the earliest stages of oogenesis in red, we saw that this fluorescence intensity decreased in each of our lines. And you can actually quantify statistically the difference here. So if you could look at the ratio of fluorescence intensity in stage 1 compared to region 2, you see it's 1.72 in our control fly, so there's more staining seen in the earliest stages of oogenesis than there is in region 2. And then you see in our heterozygous mutant flies a reversal of that. So it seems like all the staining is in region 2 and in potentially these Wolbachia aggregates, and you see much less staining in later localization to what would be the developing oocyte. All right, and these aberrant aggregates that we observed in these mutant lines were definitely more common in our mutant backgrounds than we observed in our wild type. So across 30 different germaria to 40 different germaria that were imaged for each of these different lines, we saw many more aggregates in our mutants than we ever observed in our controls. All right, so I've showed you that Wolbachia in these heterozygous mutant backgrounds seem to be inefficient for transmission, and that they display this localization phenotype where they are in these aggregates and they aren't localizing well to the earliest stages, the earliest oocyte. So we had several explanations for these, two which are fly-specific, based on the phenotypes that these particular mutations induce in the fly. So the first was that potentially our heterozygous mutant flies exhibit maternal loading or polarization defects, leading to altered Wolbachia titer. I should mention at this point that these particular alleles that we looked at, although they are lethal or result in sterile flies in the homozygous condition, they are recessive. And so these heterozygous mutant flies are thought to be phenotypically wild type. So in one way that we looked at this potential defect of maternal loading is by characterizing the shape and the size of different eggs that are laid by these lineages. So we counted 30 or 300 different eggs for each of these different lines, and we saw no difference in egg size or in shape and morphology. And importantly, the phenotype that I've showed you occurs prior to cytoplasmic dumping. So this phenotype of these aggregates of Wolbachia being kind of stuck in region 2 would be prior to any effects of cytoplasmic dumping. So we did not prefer this as an explanation to our data. The second possible explanation are that heterozygous mutant flies are deficient in stem cell proliferation, potentially reducing the niche for Wolbachia. And one way to characterize that is by counting the number of progeny of each of these lines. And compared to our control lines, each of our heterozygous mutant lines produce statistically indistinguishable numbers of progeny. So we did not find there to be any difference, at least in this gross analysis of proliferation of stem cells across the different lines. So this is not an explanation for our data. The last hypothesis that I'll explore with you are that altering the ratio of F and G actin might affect Wolbachia's ability to persist in the host or to enter the germline. So I'll focus on this particular line, the profilin null, so heterozygous mutants in this chickadee line. And what I'm showing you first is that if you compare the amount of profilin in these heterozygous mutant lines to our controls, you see a reduction by half. And that is not so surprising since they carry a single copy of the wild-type chromosome as well as a single profilin null chromosome. So that's one phenotype in these flies. I should also mention that Wolbachia could only be detected in 3 out of 20 of these heterozygous mutant flies. So using quantitative PCR, we can detect this significant reduction in profilin, and we can also detect that Wolbachia was only found in 3 of them. So a significant reduction in profilin correlated with our reduction in our ability to detect Wolbachia. We then went on to characterize the amount of actin that was in these fly tissues. So we dissected the ovaries of the fly, homogenized the tissues, passes them through an ultracentrifuge, so really fast spinning to separate the F and G actin, and then we quantified the amount of G actin by densitometry using Western blot's anti-actin. So what you see here is that in our control flies we can consistently detect a fair amount of G actin, but that using this method it was more difficult to detect G-actin in our profilin null heterozygous mutants. So there's a statistically significant decrease in the total amount of G-actin in these flies. Okay, so if Wolbachia is so sensitive to profilin knockdown, can we directly show that knocking down profilin directly impacts Wolbachia's titer? And to do that, we took advantage of the fly expression constructs and the fly short hairpin silencing triggers, and I'll explain that to you now. So we drive expression of constructs in the fly using the UAS Gal4 system, where essentially you have some promoter that drives expression of this transcription factor that then binds to the US promoter and then promotes the expression of other downstream genes. So what I'm showing you here is 2 different fly stocks. that we generated, one driving GFP using Actin as a driver. So this is something that is expressed at all times and is generally in all tissues of the host. And I hope you can see this GFP channel here where you see some green in this female. And then I'm also showing you that we can drive expression using the HSP70 GAL4 driver. So we have HSP70, which is the heat shock protein, driving expression of GFP as well. So there are many advantages to working in flies, and one of them is that there are a suite of genetic tools that we can take advantage of, one of which is this large library of short hairpin RNA silencing triggers that was developed by the Paramon lab. And we found one that was specific for profilin. And what we did is we looked at, we generated, these flies that were driving expression of this short hairpin RNA silencing trigger from HSP70. Then we shocked these third instar larvae for 37 degrees for 10 minutes, and then we harvested the pupae for RNA and DNA to see whether or not by knocking down profilin at this stage could we see a Wolbachia titer defect in the pupae. Importantly, I should mention, heat shock did not alter either profilin or Wolbachia titers in the maternal line. So in the absence of this driven expression, we don't see any effect of heat on Wolbachia or on profilin expression. And what I'm showing you here is in this HSP70 GAL4 profilin knockdown, the expression of WSP, so Wolbachia titers, here on the left, and the expression of profilin either in the control condition in blue or after heat shock in red. And I hope you can appreciate that there's a statistically significant reduction in the amount of Wolbachia we can see in these pupae and in the amount of profilin directly after we induce the short hairpin silencing trigger. So again, pointing to a direct interaction between the amount of profilin that's there, its effect on the cell and the host, and Wolbachia's ability to proliferate in these tissues. So to get to the final explanation for this phenotype, we believe Wolbachia is sensitive to the regulation of actin during development, and this can affect titer and also maternal transmission. So in summary, for the first part of my talk, Wolbachia are inefficiently transmitted in the chickadee and quail heterozygous mutant flies. The flies are otherwise phenotypically wild type, and these are recessive alleles, so they have no other phenotype besides this Wolbachia-specific transmission defect. Wolbachia that are present in these mutants are aggregate in region 2 of the germarium, and they don't populate the first egg chamber as efficiently as our wild-type controls. So in this first generation, they show a maternal transmission defect. And this inefficient phenotype is correlated with Wolbachia titers in the whole animal, the amount of G-actin in the ovary, and profilin expression. All right, so here we're getting to the second part of our talk, where I'm going to tell you a little bit about our crazy forward genetic screen in Wolbachia, for our selection for suppressors in a mutant host background. So here's the idea. Wolbachia are inefficiently transmitted in chickadee and quail heterozygous mutant flies. I've showed you that data. So the question was, can we use these host backgrounds to bottleneck Wolbachia and potentially select for suppressors of the host phenotype? So obviously, I've said this before, but Wolbachia are obligately intracellular. We cannot currently genetically manipulate them. So this might be one way to generate mutants and to also mechanistically dissect what Wolbachia are doing in different host backgrounds. All right, so for an obligate intracellular bacterium, the host is the ecology of that organism. So if you alter the host ecology, let's see if Wolbachia can adapt to that ecology. So in this first experiment that we performed, we actually passaged Wolbachia for 3 generations in this heterozygous mutant background. And then we looked at various different phenotypes for Wolbachia after passage. So there were 20 different lines established that were mutant and 20 control lines that we simultaneously passaged just to determine whether or not we see any effect of passage on Wolbachia outside of the context of this mutant allele. At the end of the experiment, we had 9 surviving and infected mutant lines, so we kept 9 control lines as well, and that's the data I'll be focusing on for the rest of this talk. Okay, so just to give you a sense of, Wolbachia's phenotypic adaptation to these lines, I'm going to show you some data that are interesting but difficult to interpret. So I mentioned before that we use WSP, this Wolbachia-specific surface protein, as a reagent in the lab. So it is an antibody to Wolbachia surface protein that recognizes Wolbachia specifically, and also can be used to look at titer of Wolbachia in different tissues. And we just have it in the lab because it's a reagent. We don't actually know, the Wolbachia community, I should mention, doesn't know what WSP does. It is on the surface. It is known that in infection by filarial nematodes of mammals, you'll get an immune response that results from WSP, but exactly what its function is for the bacterium, that is a big unknown. So what I'm showing you here is that when we dissect the flies, we can pull out the ovarian tissue from females, and we can run a Western blot with ovaries, that's what I'm showing as the O, and then what the rest of the leftover tissue we refer to as the carcass, or C. So normally in our control flies and in our first generation in these different backgrounds, we see that WSP is actually only present in the carcass. So it seems like Wolbachia are expressing this protein in a tissue-specific way because, as you already know, Wolbachia are densely colonizing the ovary, but they are not present, they're not expressing WSP in that same tissue to the same extent that they're expressing it in the carcass. And this story changes when you introduce the second generation. So in flies that continue to be infected with Wolbachia after 2 generations of passage, we now see Wolbachia upregulating this expression of a protein, this WSP, in this tissue, in the ovary, whereas before they weren't expressing it there at all. So this is one instance of a phenotypic adaptation of Wolbachia via passage through these different lines. But as of now, it's difficult to interpret what this means, except that Wolbachia is phenotypically altering its behavior with regards to these different host backgrounds. I did already show you lots of data, and hopefully you agree with me that Wolbachia are decreased in titer in these chickadee mutant flies compared to control flies. But here are the raw data to remind you of that. So what we wondered then is, could we find Wolbachia after passage for 3 generations through these fly backgrounds that recovered the natural or the control titer levels? So what I'm showing you here is relative quantitation of Wolbachia using quantitative PCR against this Wolbachia-specific gene. Here are our control flies, and here are our 9 heterozygous mutant 3rd generation flies. And what I've highlighted with the asterisks are those lineages in which we saw the low titer of Wolbachia that we normally associate with these chickadee backgrounds. So not so surprising, really low titer because of the host mutation. But then, for 4 lines, we saw a recovery of Wolbachia back to control titer. So they are statistically indistinguishable from control at this point. So in only 3 generations of passage, these Wolbachia have figured out how to make a comeback. It also turns out that these Wolbachia mutants are more pathogenic than their predecessors. So compared to their the maternal line or the progenitor line, these mutants have a more severe phenotype in the host. And what I'm going to show you now are the classic crosses you do in Wolbachia, where you cross an infected male to an uninfected female, and then you look at the number of progeny that are produced. This is the cytoplasmic incompatibility phenotype. So what I have plotted are number of progeny and our total number of progeny in our Wolbachia infected mutant line. So this is our mutant Wolbachia in this background. And then we also cleared these males of a Wolbachia infection using tetracycline and re-inoculated them with their microbiome. So we're looking at Wolbachia-free males in the same genetic background. And you can appreciate that there is a significant increase in the total number of progeny when you remove Wolbachia from this cross. And that this was specific to females. So the number of female progeny produced by this Wolbachia-infected Drosophila was much less than produced when Wolbachia was removed from the equation. So again, suggesting there's some genetic interaction between the chickadee allele and these mutant Wolbachia with regards to cytoplasmic incompatibility. So the obvious thing to do at this point, since we have some phenotypes, some mutants that seem to recover and now can colonize these mutant hosts fine, we sequenced the genomes to see whether or not we could find any differences that were in these mutant lines that could potentially explain the titer increase or the expression of WSP. And so what we did is dissect ovaries from these mutant lineages and control lineages as well as homogenize them, selected for Wolbachia using differential centrifugation. And then we also sequenced the maternal progenitor to determine the ancestral genome composition and to make sure that any of the mutations that we observed in our passage experiments weren't there previously. So here are just some sequencing statistics for each of these different lines. We created 2 libraries from the maternal stock, and I should mention these are the exact maternal flies that were used in this experiment, so they are the moms. And we sequenced 4 control lines as well as 4 mutant lines. You can see that we have a pretty small fraction of the reads mapping to the Wolbachia genome, and we used the WML published scaffold as a reference, and we used Bois and Bowtie to map these reads. I should also say that even though we have a really small fraction of reads mapping, we got 20 to 60x coverage for each line because Wolbachia genomes are quite small, so it's pretty easy to actually get good coverage even with small numbers of libraries. Okay, so we also set some pretty significant thresholds for what we consider to be, quote, real. So we set high quality thresholds for all of our reads before we mapped them, so a Q30/80. The indels were supported by reads going in both directions. Each position was covered by at least 20 reads, and we looked at SNPs that appeared at more than 8% frequency. And genetic mutations of interest were validated then by Sanger sequencing. So we'd actually still had these flies around, these particular lineages, so we were able to go back, extract DNA, amplify this region, make a clone library, and sequence 96 to determine if the fraction was similar to what we had observed in the read depth profile. Okay, so this stuff was published in AEM pretty recently, if you want to check out the paper, but I'll just highlight some of the differences that we observed. So first, obviously, we observed changes in our ancestral lineage that were not present in the WML reference. And that's not very surprising at all. So the organisms you work on in your lab are not the same as the type strain that was sequenced and is published in GenBank. So, due to just bottlenecking and random effects of passage through the lab, you will find differences. Whether or not they are, important to your particular organism is dependent on the scenario. But we found a fair number, highlighted in gray here, of mutations that were fixed or nearly fixed in all of our mutant lines and in our ancestral stock. So again, this is something that is just background mutation that was present in the ancestor. And then in our mutant lines, but not in our control lines, we saw a statistically, or a significant enrichment, I should say, of particular alleles of non-synonymous changes across a large set of different genes. So I'm listing the genes here, all the way on the left of the screen, and then for each of these mutations in red, these are non-synonymous changes, I list the frequency with which we observed these particular changes in that library. So some of them occurred at upwards of 22% frequency, which is extraordinarily high. So it was exciting to us that at only 3 generations of passage, we could enrich for particular mutations in this pool at that high frequency. The other thing that you might notice is that there is absolutely no overlap in these different mutant lines. So we did not find a single instance of a specific gene being enriched for, or a specific allele being enriched for across multiple mutant lines. And that was disappointing at first, but we did only do this for 4 mutants, and we also only allowed them to be passaged for 3 generations. So that's a potential explanation. If you're doing a suppressor screen, in E. coli, you might exhaustively sample your suppressors to be able to get a better statistical sense of whether or not you've captured all of the potential mutations that lead to the phenotype. And finally, I should mention that a lot of these open reading frames, these genes, these predicted genes here, for which we find mutations, are genes of unknown function. So it is possible that we are hitting the same pathway in Wolbachia, but we don't know it because these are uncharacterized genes as of yet. So these were the data that we presented, and we're excited about the prospects of this, if only because we figured out a way to stochastically segregate or select for mutants in Wolbachia, and if we can passage them for longer and enrich for a higher fraction of these alleles, we could potentially isolate penetrant mutants, nearly penetrant mutants in Wolbachia, and figure out what's going on with regards to these phenotypes. So we're repeating the passage for a very much longer number of generations. We're currently at F5, and we're doing it for a larger number of lines, so 50 lines for control and mutant lines. We're going to sequence a larger number of suppressors at the end of this experiment and see if we can't find any similarities across the different suppressors, and identify the trajectory of alleles during experimental evolution, classic evolutionary experiment. So please stay tuned, and hopefully we'll have more for you next time. And with that, I'd like to acknowledge the members of my laboratory, what I call the Wolbachia crew, highlighted in red. People who significantly contributed to these data are Kathy Sheehan, Marianne Martin, and Sasha Savitsky, and as well as our collaborators and the funding that supported this project. And with that, I will take your questions.
All right. So Irene, if you drop your screen down, we can ask questions to your face, I guess.
No, it's better yet.
So we don't really have any questions on Twitter except for one that Jen, I think, got answered.
Okay.
Is there anyone inside the Hangout right now that wants to ask some questions? And if you do, you can just unmute yourself and speak up.
Yeah. So Irene, oh my God, why do I have a beard on? I'm gonna change that. So my question was just thinking about these aggregates, right? The thing that you see these cells getting into a larger clump.
Yeah.
I mean, it does kind of tie in with this idea that there's something in the cell membrane going on that they're maybe even more sticky. But I was wondering, you were saying that doesn't disrupt egg development, but wouldn't that disrupt cell communication in the fly line and as it's developing? But you said, like, you were kind of answering that you don't see any downstream issues.
So you're wondering if the Wolbachia aggregates themselves would be affecting oogenesis.
Yeah, and just sort of disrupting the fly host completely.
So we don't have any evidence of that, at least based on this. Like I said, this is a crude phenotype. Counting the number of progeny and looking at egg shape and egg size and other kinds of fecundity metrics. We didn't have any evidence that the Wolbachia were doing that. That said, I have no idea if these aggregates are live. So I have Wolbachia staining, but I can't tell you that these bacteria are doing anything. And it's unclear to me at this point. We don't have. We'd like to say, yeah, this localization is characteristic of Wolbachia infecting the somatic stem cell niche, which they're known to infect in other backgrounds. But I can't tell you that's what that is because we have no specific staining for that region yet. So they're kind of just an observation, an anomaly compared to our controls at the moment.
So I was wondering about this craziness with not being able to map reads back to the WMEL genome. And you might have said this, but I could have missed it. Did you try assembling the sequencing reads, kind of like
it's not. Yeah.
From a metagenomic perspective, almost?
The reason that we can't map them is because the majority are Drosophila. And that's not that surprising because you have this enormous host fraction,
Sure.
Lots and lots of nuclear genomic material there when you do your preparation. And so people, so in the intracellular symbiote world, there's lots of strategies to try and get better resolution and enrich your symbiote fraction better. The truth is that sequencing is so cheap that it's actually pretty straightforward to just power through it. And
Right.
I guess what we're showing is that you don't really need to do really elaborate lab protocols in order to actually get enough data to say something.
Yeah. I guess I was just wondering if maybe like a metagenomic type assembly with some binning, a la Banfield Lab, Brett Baker, those kind of guys might get you further with longer contigs that you could do something with. I'm not sure.
So we don't really need contigs since we're just mapping it to a reference. So that makes it a little bit easier. But I can't, I'll tell you that the stuff that does not map to Wolbachia, the vast majority of it maps to the Drosophila genome, and we do get some random other microbiome DNA members in there, but they are not significant.
Do you have a sense of the percentage of reads that are Wolbachia reads that did not map to the WML genome? Or the reverse?
Yeah, so if I do the reverse, then you get roughly the same fractions. So if you do a Drosophila mapping on the libraries, then roughly between 3 and 10% will not map to Drosophila.
Okay, so let's see. So you have 3 to 10% of the reads mapping to Wolbachia,
Mm-hmm.
and then how many of those reads are probably Wolbachia that aren't mapping to the existing genome, though?
I would say very, very few, very, very little. And it's hard for me to say, like, if there's some reads that cover a repeat region that we have difficulty in the assembly itself. I can't say. It's also possible that this particular strategy we took. Obviously, we're identifying indels, we're identifying potential new junctions, but we have a harder time because of the short reads identifying things like really large new insertions, right?
Or rearrangements, or yeah.
Yeah, so I mean, we can identify some of the new junctions and rearrangements, but we can't identify things that are completely novel. And that said, I'm not sure. Yeah, I mean, it's an interesting thing to look at for these 3 generations. I'm not sure if we'd find it or not, so it's something to put on the docket.
Yeah, I mean, the reason why I bring it up is just because you made this great point about the fact that your strains aren't going to be very similar to the type stream to begin with. And so some of the things that you find, I mean, you could have big genomic changes with these bottlenecking experiments, especially that are altering your strains before you even put them through this whole process. And so
yeah, so how different is our maternal progenitor to WML, and did that somehow make it more likely to do one thing or another? What I can say is that people have been thinking hard about the ancestry of Wolbachia and all the different Wolbachia that exist within the Drosophila lineages, and our strain, based on the SNPs that we find and the particular, I don't know if you want to call it multilocus sequence typing, but the areas that have been annotated for these different lineages of Wolbachia, our strain is the WML type of Wolbachia. It's not any other more interesting type.
I see. Yeah. Cool.
But isn't the joke of the week how much maps to platypus?
That's true. That's right.
Okay. There's a question online from Caleb. It says, wondering why 8% cutoff for SNP finding in BRESeq was used. Did you find others that were lower, and it could be with more passage?
Absolutely, we found stuff that was really low frequency. And when you get into that really low frequency SNP, there are 2 problems with it, at least from the perspective of publishing this paper. One is that they're so low frequency that it becomes difficult to confirm it with any other sequencing technology. And they're kind of so low frequency that they are close to the level of noise. So I don't know if it was worth, I mean, I kind of made it an administrative decision that it wasn't worth spending the time to try and confirm these alleles, since it was going to be extremely difficult to do. I mean, so think about it, for the reads that we, or the SNPs that we confirmed, we're talking about 10 to 20% penetrance. In a clone library of 96, you can find them. How far was I willing to go in trying to find these SNPs that occurred at 1% frequency? So I set that threshold, definitely thinking about our ability to confirm the presence of these mutations. But it is true that we found other low-frequency SNPs, and it's also true that other people in the field, such as Wolfgang Miller, have been thinking about Wolbachia heterogeneity within individual hosts and have found really, really low-frequency SNPs using amplicon sequencing.
Cool. Are there any other questions right now for Irene?
So since you and Cameron are online right now, do you want to have a lively debate about the ultimate, the real alpha proteobacteria, if SAR11 is king and Wolbachia is queen?
I don't know, can't we both rule?
Well, yeah,
I like that you use them different genders.
That's right. There you go.
Genders. Yeah, I guess we need to gender wars in single-celled prokaryotic. I said the word. Yep. I'm on the record.
The word. I actually love
All
that
right.
word.
I'm on the record in publications saying that word too. And if two domain theory starts to hold up more, then that's just going to become an even better word with every passing year.
There's one more question that just came in. Any cool low-frequency things that you predict will be real or increase with additional passages that you would make the admin decision to go after?
Yeah. So like I said during my talk, a lot of these changes that we observed were in proteins of unknown function. So it's hard for me to say that they're cool. Certainly, some of the changes that we observed that are high frequency, that met our threshold, were changes in genes that are predicted to be involved in interaction with the host. And so we highlighted them in the paper to some extent. So proteins that have so-called anchoring domains in them that have been long assumed to be involved in the interaction with host, or proteins that have previously characterized actin interaction domains, which could potentially be of functional importance. But I think it's very premature at this point to say that these mutants are actually, or these Wolbachia mutants are actually responding to the host specific allele, because we have a combination of things happening. We have this stochastic segregation because we are bottlenecking them so severely, and we have potential selection for suppressors that can make a comeback and actually recolonize. So I would say that I'm excited about stuff that'll come out of this newer screen with a larger number of generations where we sequence more and can determine, if I see it multiple times in multiple lineages, I will be more happy with it.
Great. Okay, I think that's it, unless anybody has any last-minute questions. But Irene, thank you very much for a very enlightening seminar. We appreciate your time and all the content and for sharing with us. Let's see, I guess our next seminar is going to be in March. So check out the micro seminar webpage for upcoming schedules, and we'll see you guys next time.
Okay, thank you so much.